next one is my co-chair, Julia, how to say that? I mean, Gerasimenko.
Thank you very much. I would like to thank the organizers for this opportunity for me to speak and present our recent data. So as you can see, I'm going to talk about coronavirus today. COVID-19 was the global pandemic for over 2 years. In these past 2 years, we have learned many lessons. And one of these lessons actually, it's that SARS-CoV-2 can induce or trigger and be involved in many inflammatory diseases such as acute pancreatitis. There are many papers in past publishing that SARS-CoV-2 is associated with acute pancreatitis cases. But what exactly we know about the SARS-CoV-2? It enters the cell in the SARS-CoV-2 enters the cell by the receptor-mediated endocytosis using the S1 subunit 1 protein over here to bind the ACE2 receptor on the host cell. And so it does for many cells in the respiratory system, in actually nervous system and digestive tract, including the pancreas. And so during the pandemic, when we were restricted to access our labs, so Oleg and I were thinking, so how we can really investigate anything on the mechanism, and so which cells can be targeted in exocrine pancreas by SARS-CoV-2, and specifically what is the binding actually of the S1 and what it does to pancreas. Taking advantage of our novel method to investigate cells in freshly live isolated pancreatic globules, we actually for the first time were able to show that S1 protein can induce calcium signals specifically in pancreatic macrophages, shown here on this image, and also in pancreatic stellate cells. So this method allows us to look at many different cell types at the same time and follow calcium signaling or nitric oxide signaling in the lobules. This is the immunostaining of pancreatic macrophages, which is the resident macrophages, macrophage here, by the immunostaining with the fluorescent targeted antibodies against the surface proteins. So what we found that S1 at concentrations starting from the smallest 70 nanomolar up to 600 nanomolar, so it induces either single or train of spikes in macrophages as well as in stellate cells. Interestingly, that pancreatic acinar cells, they do not produce any calcium signals to S1 in the time frame of these experiments. These actually responses which are produced in a dose-dependent manner must be very important into the propagation of the inflammatory response in pancreas. So what we also found very interesting in fact that macrophages responded to S1 with a substantial delay and as compared to the stellate cells. You can see this delay was 4 times. So since we apply S1, so macrophages 4 times longer thinking about what is around. So that would be identification of some indirect action of S1 on macrophages. And we decided to dissect this particular question further. So we pre-incubated the pancreatic globules with the inhibitor of interleukin 18. This is interleukin 18 binding peptide. And as you can see, all these actually responses in macrophages to S1 are completely abolished. However, responses to S1 in stellate cells were unchanged. These results suggest that we can, well, propose this, that stellate cells are affected by S1. They produce the cytokines such as interleukin 18, and this cytokine acts on immune cells and they respond also to S1, but indirectly. We dissected to another point of this investigation, so to understand if calcium, extracellular calcium, has any effect on these kind of responses in stellate cells and macrophages. And indeed, in this traces, you can see in the absence of external calcium, there was no responses in pancreatic macrophages in stellate cells. But all cell types are actively responded to. For example, ATP is a typical response in pancreatic macrophages, and bradykinin is the usually calcium response in stellate cells. So previously we have proposed, as Ola already described nicely, we have proposed to crack pancreatitis using inhibition of calcium entry by specific RI1 crack channel inhibitors. And we used by that time GSK compound produced by GlaxoSmithKline, GSK 7975A, which has nicely blocked main toxic effect of non-oxidative alcohol metabolite DOEE. Finally, so we found that cell necrosis was dramatically suppressed. And this actually protective mechanism was developed further. So we had confirmation of in vivo experiments using the same blocker. And a novel blocker was recently proposed by actually Calcimedica. This is the CM4620, and it's named Xora. And as you can see, so that's the reality at the moment. So it's used in human trial for the acute pancreatitis side effect from pediatric treatment of acute lymphoblastic leukemia, acute pancreatitis with severe cases, and also COVID-19 pneumonia. So this is a kind of promising step forward. And we use the same CM4620 Xora in our experiments. We use the pretreatment of pancreatic globules with 10 micromolar CM4620, and we found that there's a complete blockage of these responses in macrophages, and in 75% of stellate cells also responses were blocked. However, the rest of responses in stellate cells were significantly suppressed. So that's the summary of our investigation. So first of all, for the first time, we have shown that S1 elicits the calcium signals in both pancreatic stellate cells and macrophages, but not in pancreatic acinar cells. Stellate cells appear to be a primary target for the S1 proteins, and we propose that they might release the interleukin 18, that in turn activates macrophages. And so we found that the blocking peptides, the IL-18 binding peptides, selectively eliminate calcium signals in macrophages. So why it's important? So it's a very important step of this paracrine signaling within the tissue when we have indirect effect of S1 on macrophages, but it looks like S1 primarily targets stellate cells. ORI crack channel blocker CM4620 suppresses calcium signals in both cell types. And so we can really predict that this is a very important potential therapeutic target for treating inflammation in general, but also in reducing the risk of acute pancreatitis in mild cases in patients with COVID-19. And I would like to thank my collaborators from Cardiff University, from Bogomolets Institute, also Jinan University, and Great Ormond Street Hospital, London. Thank you very much for your attention.
Thank you very much. We have time for a few questions. I would handle this, so maybe I'm not that fast. Try to.
Very interesting observation. So can you model this in like an experimental pancreatitis model, like adding S1 would make things worse, like in a simple cerulean model?
Yeah, that would be great, actually. We just started because lab was recently open. So yes, we have many plans, and this is really good suggestion to try to see what's going on in the model situation. So we have in our lab two quite important models, so alcohol and also asparaginase induced. So we can try.
Just a quick question. In humans, the viremia is actually fairly short-lived, and it's the immune response that seems to drive a lot of pathology, especially through IL-6. And I was wondering if you had how you work that type of combination of things into your model.
Well, so of course, this is just the beginning of the project. Yeah, we can try different cytokines to also to investigate. It's also very important to understand so how pancreatic isthmus cells may be involved later on in all this response, both of that inflammatory, of course, response. So that would be very interesting.
If I can ask a last question, that do the same things happen in the lung?
Well, we are not expert in lung.
Yeah, I know, but probably you look at the literature of that. So
Right.
since, I mean, the primary inflammation is usually happening in the lung, I mean, in COVID.
Yes. Yeah. I don't know really.
Okay, because this may send a message to that direction too, to this kind of stuff. Okay, thank you very much. Thank you.